Following this, we have Professor Bernard Wood to speak to us on where does the genus Homo begin and how would we know? Bernard.
It's a pleasure to be here. Thank you, Fred, for asking me and for being a splendid publicist. The check's in the mail. And if you want Fred to introduce you, I'll give you his email address. This is the first time I've ever spoken at a meeting with both Richard Leakey and Philip Tobias. The former is my sort of scientific father, and lest you think that Philip may have been guilty of some indiscretion, if you look at our profiles, you will realize that there might be something in that. But actually, it's an example of homoplazy. The chins we have, we did not inherit from a recent common ancestor, and I'll be talking about homoplazy a little later. But this lecture actually does give me an opportunity to pay in public, or to say in public, how grateful I am to Richard for the opportunities that he gave to me when I was a young medical student. And the message I want to give to all you guys out there who run expeditions is that if some sort of callow youth writes you a letter, or at least if your professor tries to speak to your mother to persuade you to take a young person on an expedition to northern Kenya who you've never known, you should try and say yes, because that's what Richard did. Well, that's what Richard did. I know he's regretted it ever since, but nonetheless, that's what Richard did, and I'm immensely grateful to him for that opportunity, and I want to use this opportunity to make that my gratitude, or to thank him in a very public forum, and also Meave, because she was also instrumental in making sure that Richard didn't sort of boot me off the team when I was thinking things that weren't maybe. Yeah, absolutely, Richard, absolutely. But just for the public record, I did not introduce Homo rudolfensis. Mercifully, I didn't have to, because a Russian called Valery Alexeyev had already made 1470 the type specimen of a species called Pithecanthropus rudolfensis, and of course it was no longer a sensible thing to use the genus name that he had used, but nonetheless it wasn't me. And I certainly, even though I'm 62 and have grey hair, Richard, I would not do anything that you didn't want me to do. So let's just make sure that I wasn't a bad boy. I might have been a naughty boy, but I wasn't a bad boy. Okay, so what we want to do, we're looking at a very small part of the life here. It's just a little bit of a twig here, which has modern humans on the end of it. And what we're interested in is how far back down that little twig that may have a few branches in, how far back does the genus Homo go? Now, in this diagram, which has been color-coded for the people from New York, you can see here these are all hominins that have been included in either in the 4 species which have been proposed as the earliest hominin. You must realize that you can get the fossils for all those hominins very, very easily in a carry-on suitcase, and you'd still have room for a bottle of whiskey and your shaving kit. Okay, so there is not a lot of evidence for all those things, and to my mind, I don't think any of them is a dead cert early hominin. That's just to try and get the discussion going. These are what you can call archaic hominins. These are the ones that have really large molars and premolars, and these are ones that have molars and molars that are less large. Your first mandibular molar is about the size of your little fingernail. That's just a fact. If you bite your nails, then that's your problem, but it's the size of an unbitten little fingernail. The first molar of OH5, or Zinjanthropus, is about the size of your thumbnail. And the first molars of these creatures here are about the size of the nail on the end of your index finger. So I just want you to remember that. So the question is, does the genus Homo go down and include Homo habilis or Homo rudolfensis? And to be honest, I don't know. I'm not sure, and certainly have said in print, although my colleagues, they never read what you say, and so they take absolutely no notice of it. All I've said is that of the hypotheses that you can put forward about the taxonomy of early Homo, it seems to me slightly more probable that these are two species than they are one species, but it's impossible to reject the hypothesis that they're one species. I mean, I've said that again and again and again, but it makes absolutely no difference. Everybody thinks I'm completely committed to the idea that these are two species. So the question is, should Homo just come down as far as Homo ergaster? And if you don't like Homo ergaster, you can call it early African Homo erectus. And I think one of the interesting things is that new specimens, and I think Richard was alluding to this, have rather sort of blurred the distinction between Homo ergaster and Homo habilis. And the question is, have they blurred it to the extent that there should not be a distinction, or is there still some distinction that can be made? Or might the genus Homo not even extend that far? You can make the argument, it seems to me, that as soon as Homo was able to develop language, a complex language, that was an enormous move out of one adaptive mode into a different adaptive mode. And if you were just looking at the adaptive level or the adaptive zone of these early hominins, you can make an argument that it should be restricted to Homo sapiens. There are people who think that actually this whole lot all belongs in one species, but they're barking mad. And you should not waste any time. You should not waste any time thinking about that. So we suggested a few years ago that the definition of a genus should be either a species or a monophyletic group, and I'll tell you what that means in a minute, whose members occupy a single adaptive zone. So let's try and put that into ordinary English. Well, actually not that ordinary English. A monophyletic group is also called a clade, and the adaptive zone, a single adaptive zone, will be called a grade. So what do we mean by a clade, and what do we mean by a grade? And a genus needs to be both. It's not sufficient for it to be one of those things. It should be both of those things. So what is a clade? Well, you're American, and so the natural thing is to think about a definition. All of the taxa, no more and no less, that are descended from a recent common ancestor. But let me try and put that into American. And of course, what one needs to do is to think of motor cars, because this is America. So this is a clade. There is a common ancestor for all these motor cars, and there it is. And those motor cars, plus all the other cars made by Ford, although these days they're not made by Ford, they're probably Ford belongs to something else, and it's much more complicated. But just allow me the simplicity of the metaphor, that all these cars belong to the same clade. Okay, no matter how different they look, they all share a recent common ancestor, which is not shared with cars from another motor company. So they all share a common ancestor. They all share a common ancestor that is not shared with other makes of car. So we're looking for that equivalent in the hominin fossil record. So what is a grade? Well, it's a group of taxa that shares a suite of functionally adaptive features. Now, what's important is that if you're a paleontologist, you have to make that decision based on the fossil record. So it would be nice if they were all capable of speaking, but we don't know what the fossil evidence for speaking is. it would be nice if they're all capable of making the same sort of stone tools. But we don't know what the fossil evidence for making the same sort of stone tools is. So you have to think about adaptive grade in terms of what you have in the fossil record. So we were thinking about adaptive grade in terms of, say, the size of the mandible that you have, because we have lots of mandibles. Is the body of your mandible large for your body size or small for your body size? Are the molar teeth large for your body size or small for your body size, and so on? So we restricted ourselves to things that we could make reasonable deductions about using the fossil record, as opposed to the things that we would like to make deductions about, but we can't. So here is a grade. These three SUVs are functionally similar. They're all SUVs, and they share a common ancestor. But they don't share a recent common ancestor. The common ancestor they share, and I cannot find when the Toyota Motor Company and when the Toyota Motor Company shared a recent common ancestor with the Land Rover company. My guess is that it's way back in the early Benz cars, and so they share a common ancestor, sure, which was the earliest motor car, but not a recent common ancestor. So when we look at this group and we ask those questions, we say, are Homo habilis and Homo rudolfensis in the same clade as later Homo, as other later Homo species? The results vary. And the results vary according to the specimens that you include in the cladistic analysis, and they vary according to the variables and the characters that you include in the analysis. But to be fair, many of them actually do come down saying that habilis and rudolfensis are in the same clade as later Homo. But when you look at those results, it's not clear to me that they're really reliable. And what do I mean by something that's really reliable? Well, what I mean is that if I need a neurosurgical operation, I say to the neurosurgeon, how many times have you done this? He says, well, 3 times. I'd really like him to say 300 times or 3,000 times. And how many of those operations went well? Well, 63% of them. And you say, well, yeah, let me think about it. Let me think about it. So what I'm wanting is results of cladistic analyses which are robust in terms of, say, let's just pretend that we can't use all the character states. Let's take a random subsample of the character states, and let's just pretend that we never found Australopithecus garhi. Would you get the same cladogram if you made sort of adjustments that actually may be due to chance? So he really pays your money and takes your choice. I think there is evidence that they're in the same clade, but I also think that evidence is not as robust as I would like it to be. And this is just an example of one study, and if you change just a few character states, or if you change the length of the tree, then you can actually get a different looking tree. Why is this problem? Well, let's get back to the tree outside my office window. The problem is the homoplasy, this is morphology that I share with Philip Tobias, which I assure you is not inherited from a recent common ancestor, unless Philip's been doing something that he shouldn't have done. So this is the tree outside my office window, and if you want to reconstruct phylogeny, it's clearly a good idea to do it in the winter. Okay, it's a good idea to do it in the winter because you can see all the twigs and the branches. The problem is that we do our phylogeny reconstruction in the summer when the leaves actually obscure some of the branching pattern, and the leaves are actually homoplasy. I mean, that's the message I want to give you. It's not as simple as that, but that's the message I want to give you. And really it depends whether you think there's a lot of homoplasy or whether you think there's not much homoplasy. And that's the extent of the argument. So the question is let's ask about the grade. In other words, okay, let's assume that they're the same clade. Are they the same grade? Well, when you look, you find that whereas modern humans, your molar teeth are the size of your little fingernail, in Homo habilis, despite those being small creatures, relative to their body mass, their molar teeth are the size of the nail of their index finger. Whereas if you go to something like Homo ergaster or Homo erectus, their first molar teeth are the size of their little fingernail. In other words, like us. So Homo habilis has relatively larger mandibles than later Homo has for its body mass. It has relatively larger molar teeth relative to its body mass. And as far as I can see, there isn't a lot of evidence that it's an obligate biped. And if you put all those things together, it seems to me that Homo habilis doesn't readily find itself in the grade within which later Homo is. And that's just saying in PowerPoint just what I've said just now, okay, which is what most PowerPoint slides tend to do. So if you don't like them in Homo, you have to put them in another genus, and for the sake of annoying some of my colleagues who I know and love dearly, I'll put them in Australopithecus, because that makes Australopithecus an even messier genus than it is already. And the question is, okay, if you don't think Homo extends down this far, well, why not say it certainly extends down to Homo ergaster? Well, although Homo ergaster, all cladistic analyses have associated Homo ergaster with later Homo taxa. So in other words, if you look at the evidence in terms of a clade, this is just an example of Homo ergaster, and Richard has already shown you a picture of that. Here's another example of Homo ergaster or early African Homo erectus, and there's not really any doubt in my mind that that's the postcranial skeleton of an obligate biped. But there are some things where Homo ergaster is not obviously, so we think it's in the same clade as later Homo, but it's not obviously in the same grade. Certainly for brain size, it's not obviously in the same grade, and certainly for life history, it's not obviously in the same grade. So if you want to include Homo ergaster or early African Homo erectus in Homo, you have to make some concessions. You have to say that I'm willing to have species in there which have a still pretty primitive life history, and I'm willing to have species in there which have still a relatively small brain size for their body mass. So you then say, well, okay, if you're still skeptical about Homo ergaster, and God, you must be a skeptical guy, Homo erectus has surely got to be in the genus Homo. Well, it's not as easy as that. Why? Because of the French. And as you know, you're Americans, the French are the source of all serious trouble. Clade, sure. There is no argument that they're in the same clade as later Homo. Grade, well, this is where the French come in. A very ingenious piece of work by a French group looked at trying to reconstruct the life history of Homo erectus. There's no doubt that these are Homo erectus. And when they did, they found that the relationship between the percentage of the adult endocranial volume and the age, the reconstructed age, that these creatures weren't growing their brain like other later Homo taxa. So you can even have some doubts about Homo erectus, or you can just dismiss the French. So it might be here, might be there, it could be anywhere. There is obviously a complication with Homo floresiensis because all this discussion actually took place, or most of these arguments took place before the discovery of Homo floresiensis. And Homo floresiensis does complicate things. It was found in this cave here in Western Indonesia, and I thank Peter Brown for sending me these images. This was in the paper. This is the spit where the hominin skeleton was found, and that's a photograph of the excavation. And that's where people like to put it. They put it in Homo. But the question is, this is a pretty small creature. It's got a pretty small brain size. There is the skeleton. Some people think it's pathological, and as Philip Tobias has said, this is not new news. Every time somebody finds something interesting and new, there is always somebody willing to put their hand up and say it's pathological. Okay, shouldn't take any notice of it, it's pathological. Now, admittedly, there is only one cranium. There are other specimens, but this is the skeleton of an individual that's meant to be a microcephalic sort of dwarf with all this skeletal asymmetry, and was sort of loping around Indonesia, with a sign on it saying "Born Loser," and sort of, you have this image of Homo floresiensis, and here it is compared to a normal, inverted commas, Homo erectus. There are a couple of jaws, but there is only one cranium. And there are the kneecaps. Now, it's very difficult to find a couple of patellae that are as alike as that. And this is meant to be in a skeleton which shows all this incredible asymmetry and stuff. So I don't really buy that. And I think the null hypothesis ought to be that this is a normal individual of something until there is a lot of evidence to prove otherwise. And I've not seen the lot of evidence. And here it is beside a modern human. Some guys with a brain this size think that these have a pathological brain. My guess is that the pathology might actually lie somewhere over here and not over there. Don't write that down, Bill, please. Okay. And what's interesting that they made stone tools much like these 800,000-year stone tools from another site in Indonesia at Marta Menge. So, when does the genus Homo begin and how would we know? I've tried to indicate how we might know. That is, it should be in the same clade as Homo sapiens, and it should be in the same adaptive zone or grade as Homo sapiens, plus other extinct Homo taxa. Somebody a lot more impressive than me had worked all this out a long time ago. And Charles Darwin wrote in The Descent of Man, it would be impossible to fix on any point when the term man ought to be used. We should remember that we don't actually have to come up with an answer. We may actually come up with an answer to say that it's a very interesting and useful and an important question, but we just may be able to come up with some sort of criteria, the beginnings of a list of criteria. And that's what I anticipate that we will do in the next few days. But I want to thank the organizers, and I want to repeat my thanks to Richard. You were extremely kind to me, and without that opportunity, I would be an orthopedic surgeon driving a very large Mercedes with a house in Barbados, and I would be just about to take retirement and live in the style to which I've become accustomed. But because of you, here I am taking an economy flight between Washington, D.C. and LaGuardia. Although, thanks, Fred, I was actually picked up for the first time in my life in a limo, and I realized that Long Island has more style than I thought. So, thank you very much.
Thank you, Bernard. What I'd like to do now is to turn the proceedings over to John Fleagle, who will moderate the panel discussion. Panel discussants would please come up to the stage, Dr. Chris Dean, John Fleagle, William Jungers, William Kimbal, Marta Lahr, and Philip Rightmire. Dr. Fleagle will moderate the session, the discussion session. If there are any questions that any of you have from the audience, we request for the purposes of the taping that you please use a microphone for your question. Anybody that has a question, please raise your hand and a microphone will be passed to you. Turn it over now to John Fleagle. Thanks, John.
Thank you, Fred. My name, as he said, is John Fleagle. I'm a professor of anatomy here at Stony Brook, and we have a very distinguished panel over there who have been invited here for the workshop that's going to follow, as many of you heard. Because we have more people here than we could put on the podium in a reasonable amount of time in the day, we gave these people the opportunity to sort of ask questions, add comments to what the speakers have talked about, or raise other new points. I will let them say a few words, and then I will turn it over to the audience, and you too can address questions either to these people or to some of the speakers that you've heard this morning. So first I'd like to ask each of you to sort of introduce yourselves, and then if you have a comment, we'll do one by one. Why don't we start with Chris Dean?
Okay. My name's Christopher Dean. I'm a professor of anatomy at University College London. Listening to the speakers this morning, I think the thing that struck me most was the account, the historical account of both the discoveries and the descriptions of fossils. And increasingly I was beginning to think how science works. It really does work. And most of my colleagues are experimentalists, and they go to the lab, they do experiments, and the results of that experiment drives the next experiment. And they may get there in a different way. They may think that their form of science is different to ours. But actually, in paleoanthropology, it's quite clear that we get the right answers in the end. This iterative process of reflecting and looking back at what we do. The evidence doesn't come in the sequence which we might like through experiments. It comes in a rather sporadic way, in an uncontrolled manner. But ultimately, through the kinds of processes of asking the sort of deductive questions that we've been doing, we get to the truth. And I think that's perhaps what Richard certainly alluded to in his talk, and what's really happening.
Bill?
Yeah, I'm Bill Jungers. I'm a professor in the Department of Anatomical Sciences here in the School of Medicine at Stony Brook. Some of you may have been wondering, sure, a lot of heads being shown today, and I'm kind of interested in the bodies of some of these animals. We didn't hear much about them yet. I have a feeling we're going to hear a good deal about the bodies this afternoon with Dan Lieberman. But there are some important postcranial remains that bear on all these issues that I hope will be introduced in the workshop. And although no one ever really needs to speak for my colleague Randy Sussman, I would note that the issue of the postcranial remains, including the hand and foot and their juvenile status, I think he's put together a fairly compelling case that all makes sense as a single individual. We can argue about OH35 at another date, but I think he has good developmental criteria to suggest that's within the range of variation of a single individual. So I don't think we have to worry too much about that one. However, the thing that strikes me, compared to this richness of crania, we have remarkably few associated postcrania that include limb elements that are complete enough to look at the kinds of things that I'm personally obsessed about, and that's limb proportions and their role in locomotion. And it's this vexing, either it's a curse or convenience, that so many of these are fragmentary, and they sort of lend themselves to almost any interpretation. But the fact is we have relatively few specimens that are complete enough to actually say something about body shape and limb lengths in general. And that's why one of the specimens that you saw briefly, the strapping youth from West Turkana, is so important in this story. Here we have an individual where we can get a sense of what the body plan and body shape of early Homo was like. And we really can't do that anywhere else that I'm aware of until maybe we hear something about Dmanisi in the next millennium. The thing about the Homo erectus or Homo ergaster specimen that really intrigues me is this the body design that got out of Africa, or are we looking at the body design that evolved in Africa? I think we have to start thinking about ecogeographic variation, even in the earliest genus Homo. I don't think we have a sense of what body proportions and limb proportions really look like at this stage in human evolution. If we are a polytypic species ourselves, I see no reason to believe that Homo erectus or ergaster might have also been polytypic and related. And there may have been differences related to where they lived and what they did. And again, the material that's as old as any of this stuff from Dmanisi is going to play, I think, an important role in helping us understand that. And again, I'm intrigued by Richard's comments about pathology, or rather, I guess Philip's comment about pathology playing such an important role. That's our first response is if it's different, it's weird, it must be pathological. And I'm certainly currently embroiled with my colleague Susan Larson in the description of this Homo floresiensis material. And I'd like to run a few things by my colleagues this week and be happy to answer any questions you might have on that as well.
Other Bill?
I'm Bill Kimbal. I'm a professor in the School of Human Evolution and Social Change at Arizona State University. And I think the overriding comment that I would like to make, especially to those in the audience who do not try to make a living in this field, is the idea that many of the debates we have about the path, the pattern, the causes of human evolution are not always about the details of the anatomy of fossils. There is certainly that aspect to it. Without the fossils, we'd have nothing to talk about. However, each of us, and you heard three very different perspectives this morning, all of us bring to the table a different set of perspectives on human evolution. And they inevitably color our perception of the fossils and their anatomy. I don't want to be accused of being a relativist because I, in many ways, abhor relativism. There is truth out there, but the question is, what are the best methods to gain the true story of the emergence of the lineage that includes animals who are more closely related to those of us in this room than they are to an Australopith, for example, A. boisei. And it seems to me that in order to get to that point, one of the things we need to do is sort of peel the onion, as they say. And I hope in the workshop we'll be able to do that, is reveal the biases we have, to sort of put aside the fossils in some cases and talk about how we think the process of evolution has played out. The human lineage is not different from any other lineage of animals that have evolved on the planet, at least to the point where populations of our ancestors became more dependent on cultural means of dealing with the environment than strictly biological ones. And at the origin of the clade, or the group of organisms who share a more recent ancestor with you than with Zinj, we're not dealing with animals that are inseparable from their environment in any meaningful way. That had yet to happen. And so the question that we need to address is, first, it seems to me, how do we uncover the earliest traces of those populations? And we have to create a set of expectations, it seems. We have to use what evidence we have and look backwards with it and try to make predictions, testable predictions about what we expect, given our biases, to be the biographical profile, if you will, of the earliest populations that share an ancestor with you and me. And then ask questions about why or how those changes occurred. And so we have two separate tasks that are interrelated, but from an epistemological or methodological point of view are separable. One is based on an analysis of the fossils given our biases and an attempt to identify those elements in the fossil record that seem to indicate the beginnings of this group, this lineage or group of lineages. And then the second is to ask questions about process in relation to local and regional environmental changes, etc. And I hope that we'll, in the coming days, when we sit around, we'll be able to make some headway on both of these issues. But they are, I think, the overriding message from my point of view is, number one, we bring our biases to the table when we look at the fossils, and number two, we have two different tasks to perform. One is to identify that population of the organisms more closely related to you and me than to Zinj, and the second is to ask how they achieve that status.
Hello, my name is Marta Lahr, and I work in Cambridge in the UK. I actually was thinking here, I might be saying exactly the opposite as Christine, so I should watch it. I enjoyed the talks this morning enormously. And yes, in science, one does get to the truth. But in paleoanthropology, it is slow. And that truth is not, even when we think we know, we still might get a surprise and find out that we didn't know. I actually was most impressed by listening to Philip and thinking, how much has the sequence of discovery actually shaped the interpretation? So we create references, and we create taxa, which then we have to stick to those names forever. And then forever, we're referring back to perhaps a fragmentary fossil, or perhaps the not the best representative, or maybe the weird one, or maybe an off lineage. So the actual history of discovery has shaped a lot the way we interpret it and how we build the story and why we argue so much. And then Richard was saying, well, can we look at them just forget when they were discovered, forget which got the name first. And that, in my view, would be an essential thing to do. The other thing I was very, perhaps shocked, was with Bernard. Yes, maybe we cannot point down exactly when a genus begins. I don't work in the Pliocene. I have a confession to make. I have never personally seen a Pliocene fossil or been to a Pliocene site. So this is not my field, and I am not intimate with all the material. And yes, but in my particular specialty, which is much later human evolution, we cannot even pinpoint the beginnings of a species. So the beginnings of a genus, of course, it will be elusive. We might change the threshold, but surely we have to look for it. I mean, if we say there is no way that we can tell, it could be between modern humans and everybody else, yes. Or it could be between Australopithecines, really, really clear Australopithecines, and everything else. Or it could be in every step in between. Okay, that's a fair comment, and it's important to keep in mind. But surely finding the origins of the genus Homo has to be exciting, and we should try to do it. So I think that we're going to have a wonderful 4 days ahead. Thank you.
I'm Philip Rightmire. I owe my allegiance to State University of New York, Binghamton, and also to Harvard University at this point. I don't want to take a lot of your time this afternoon, since I'm sure you have questions of your own. We've heard a lot this morning about early Homo, first representatives of the genus Homo, either Homo habilis writ large, or Homo habilis perhaps in a stricter sense, with Homo rudolfensis right alongside. There are several good crania, it's true, well-preserved specimens, jaws, a number of teeth, not very many postcranial bones, unfortunately, that are clearly associated. This material has been wonderfully described in great detail. You know the monographs, Philip Tobias, of course, and Bernard Wood, responsible for these very important and detailed descriptions. One has to point out at the same time that even after 40 years of work, important discoveries in Africa and now elsewhere, the samples are very small. We don't have a lot of material to work with. Because of that, I think it's quite clear that we do not, at this point, fully understand the extent of variation, the levels of variation that are probably present in these groups. Richard Leakey has put his finger on just this point, said it very well indeed. There are small and gracile specimens right alongside some larger ones. There's been a little shuffling in the dates just recently. It seems pretty clear that in East Africa there are Homo habilis-like creatures close in time to larger Homo erectus-like creatures with more expanded brains. In Western Asia, at Dmanisi, we have the same situation, but the fossils are found in one site, and indeed in a restricted part of just one site, where they are all probably of about the same age. There's a tremendous amount of variation there. So it seems to me that it's probably premature to make sweeping decisions. Surely it's too early to toss the small people out of genus Homo. I think over the next few days we'll probably reach some new conclusions, and certainly I'm all for that.
Well, thank you very much. I think everyone can see we're in for an exciting and probably unending series of discussions over the next few days. There's a lot of meat there to chew on and things to worry about. We now have a few more minutes that we can open up questions from the audience, either of the speakers that you heard from the podium or from some of these distinguished experts that you can see in front of you now. If you'd like to ask a question, I'd just say just raise your hand, and when I call on you, there are people on the side with microphones, so don't try to ask a question without a microphone because no one will hear you. Does anyone have anything they'd like to have clarified? Yes.
Hi. Listening today was very illuminating to learn from all these gentlemen about how much we don't know. And sitting here, I started to wonder, is there anything we do know that could be succinctly put, I guess? Thank you.
Anyone want to take that? Please? Bernard. You can handle that. You had lots of slides about what we know.
There is a lot that we do know. There is a lot that we do know. You should realize that Philip Tobias knows more morphology in his little finger than most of the rest of us do in the rest of our bodies. And so I think it's pretty clear, certainly to my mind, but it's obviously not clear to everybody's mind, that the Homo habilis, the non-Australopithecus boisei sample at Olduvai, has things in common. There are large ones and small ones, but nonetheless, there is morphology that they have in common. So the first question you could ask yourself is, are all the non-robust hominins from Olduvai, do they belong to the same species or not? And that would be an important start. Because although it's nice to abandon the historical appearance of sort of taxa, the historical appearance of the specimens and the taxa, you have to start somewhere. You have to sort of say, well, we sort of know this, and let's start from there and work outwards. And so I think there is an awful lot that we do know. We all sort of glossed over the morphological evidence. There is a lot of data. What we're arguing about is how you should interpret those data. Sometimes the creationists, they take the view that because we are not all completely decided and unanimous about how we interpret the data, that there are no data. That's what they say. There are data. There's lots of data. We would like more. We would like more associated skeletons. We would like better preserved specimens. But don't go away with the impression that there are no data. We just aren't clever enough to know at the moment what those data how should we interpret it. And just to get back to Marta, I'm not suggesting that we shouldn't try and find out where the genus Homo begins. I'm just suggesting that wise guy, much more than a century ago, he made the point that it's damn difficult to work out. And it depends what your criteria are. And that's the same in lots of things in life.
John?
Does that help?
John?
Probably not.
I just want to tack something onto what Bernard said. We in fact do know an awful lot about the early evolution of this group beginning at around 2 million years ago. But between 2 million years and 3 million years ago, we have a terrific gap in the fossil record. There are a lot of creatures out there, there are a lot of early hominins out there, but most of them seem to have their affinities with groups that don't include the direct ancestors of us in this room. Since 1990, there have been exactly 3, maybe 4 discoveries of fossils over that million-year period of time that seem to have a reasonable chance of bearing on the evolution of this group, this clade. That's a pitiful number compared to the task we have before us to define the origins of the clade. So what we need to do is, to improve our knowledge base, we need to be going out into the field and identifying packages of sediments that we have good evidence date to between 2 and 3 million years ago, and increase that sample. And then, say 10 years from now, get back together here at Stony Brook and have the same discussion. And if we're successful in our field work, I would guess that the discussion will proceed very differently than it will today, over this week.
Can we do it in Jamaica?
We can do it in Tempe, actually.
Are there any other questions, comments from the audience? Yes, back there near the back in the red shirt.
Hello. I'd just like to know, are you certain that the difference between 2 species molars being, say, like the size of my thumbnail and the size of my index finger actually represents 2 different adaptive grades, I guess it was put, because by my naive reasoning, it seems like they could both eat the same diet because the difference between the molars isn't that great. So are you certain that that really means that?
Where's Peter Unger when you need him? Is it here? Is Peter here?
I don't know, but Bernard, you were that was your analogy. Yeah.
Never use analogies. Michael Day, who was my professor, went to teach anatomy for a little while in Nigeria, and he was teaching the pelvis, and he was trying to give these students some sort of idea of what the ovary was like, and he said it's like a walnut. And all these Nigerian students, were presumably, what the hell is a walnut? The point of the analogy was to give you something that you could use in your mind as a visual way. There are data that would suggest that although you think the fingernail of your index finger is not much bigger than that of your thumb, and maybe your thumbnail is not much bigger than the nail of your index finger. Actually, if you look at real animals, their molars in relation to their body mass do not vary more than that. It's interesting that these variations are not that great. And when you look at the size of the mandible compared to the estimated body mass, although I would have to say that our estimates of body mass are very poor, and Bill will immediately grab me at lunchtime and say, look, the error bars of these are great. Sure, the error bars of these are great, but I'm just suggesting that as a hypothesis. And the whole thing about science is that you are allowed to generate hypotheses. You can't get these hypotheses out of fresh air. You have to have some evidence behind the hypothesis. And my hypothesis is that these 3 animals with thumbnail-sized molars and with index finger-sized molars and with little finger-sized molars are in 3 different adaptive grades. And if Peter Unger was here, it would be very interesting to see whether the microwear is different on these 3 things. It will be very interesting to note from Matt Sponheimer whether the isotope signature is different on these 3 things. So you can come up with a hypothesis, and there are ways of testing the hypothesis. So yes, you're right that they might all be in one adaptive zone, but yes, I'm right that until you change the constitution of the United States. I can come up with some hypotheses, and you can shoot them down. That's what democracy is all about.
Okay. More questions from the audience? Is everyone waiting for dinner? Oh, in the back over there, Danielle.
Professor Tobias referred to that exciting early point in the mid-70s of the claim that habilis had speech-like areas from the endocast in Broca's area and Wernicke's area. What happened to that line of thinking, and is it a lost cause looking for anatomical equivalents involved in the origin of language or speech? Could we be, if we had enough postcranial skeletons, could we be looking at vocal tract reconstructions, that sort of thing, or is this all too ambitious?
Thank you for bringing that up. It's not a lost cause. When I first suggested this in 1980, there was another hushed silence. There was indeed one of the mechanisms by which you shoot down an hypothesis is to ignore it. And that was what seemed to be happening at that stage. When I presented this in Chicago, there wasn't any discussion. I've checked out the published account. There was lots of discussion on my paper, but none on that point. And then I began to wonder, was this another premature discovery? Was it a potential premature discovery? And over the ensuing 20 years or so, more and more people who studied the original material came to be impressed. The first one was an American lady paleoneurologist in the person of Dean Falk. And in 1983, she said that it was right. She used Richard's fossils from East Turkana, and on those she found also these caps, Broca's cap and Wernicke's cap. And she was a strong supporter from then. And gradually a number of other people, Stringer, Andrews, Lieberman, and Sir John Eccles. It was nice to have a Nobel laureate on one side. He adopted this and based most of his final book when he was on his deathbed on this viewpoint. So I would say that there is a growing tide of opinion that Homo habilis, or early Homo, did have some means of communication, lingual communication, spoken language, and that it used these bumps on its brain to speak. So I don't think there is enough evidence to abandon the viewpoint. Thank you.
This afternoon's program involves broader perspectives on early Homo, ranging from archaeological perspectives on diet and ranging behaviors to the energetics of the kinds of tissues, especially brain size, that makes Homo separate from other primates, and then the aspects of our postcranial skeleton that Professor Jungers alluded to, such as the adaptations to long-distance running. So we've got quite a smorgasbord this afternoon of aspects of human evolution that weren't touched upon this morning. I'd like to take a few minutes to introduce the speakers. It gives me 5 minutes here. That's impossible. But that's all right. You'll bear with me. I take this from good heritage. Our first speaker, Robert Blumenschine, is professor of anthropology at Rutgers University and director of the Center for Human Evolutionary Studies there. He obtained his PhD from the University of California at Berkeley in 1985 and since 1989, his research focus has been directed at archaeological excavations at Olduvai Gorge in Tanzania. Rob co-directs the Olduvai Landscape Paleoarchaeology Project, which is a broad interdisciplinary undertaking that aims to sample broad landscape perspectives around 400 square kilometers, around the Paeleo-Olduvai Lake Basin at discrete temporal horizons. This work aims to provide critical information about diet, ranging behaviors, and technological strategies of the hominins that inhabited this area in the early Pleistocene. Professor Blumenschene's excavations have been rewarded with new human fossils, and one of these, published in 2003 in the journal Science, which was shown by Professor Leakey this morning, has certainly had a significant impact on our views of the hominins that frequented those lake shores. Rob's work is an exciting departure from more traditional archaeological approaches to the record and focuses on specific sites, and this approach is reaping its own rewards with a much more integrated picture of early human evolution. Rob is widely published in journals such as the Journal of Archaeological Science and Science. Our second speaker, Mark Maslin, is currently the head of the Department and Professor of Paleoclimatology in the Department of Geography at the University College London. He's also a senior member of the Environmental Change Research Center there. He obtained his BSc in Geology in 1989 at the University of Bristol and his PhD in 1993 from the University of Cambridge for his work on the paleoceanography of the Atlantic during the Pleistocene, a period of dramatic environmental and climatic change. In 1993, he was retained by Greenpeace as a consultant, and from 1993 to 1995 was at the University of Kyle. He joined the faculty at University College London in 1995. Mark's research work focuses on stable isotopes and what they can tell us about climatic change, and he's been especially interested in the onset of northern hemisphere glaciation and its association with vegetational changes. He's interested in paleoceanography, world climate, and tectonic uplift and its impact on environment. Global perspective that doesn't get much more global than that. He's published already in over 70 research articles in leading journals, and in 2005 published a paper in Science entitled The Late Cenozoic Moisture History of East Africa, which has had a profound impact on the field of human evolution, or at least the way I look at it. I first met Mark at Boulder in 2003 at a conference organized by Matt Sponheimer. I don't think I'll go any further with that. Our third speaker this morning is Dr. Leslie Aiello, who's the president of the Venner Gren Foundation for Anthropological Research, a position she's held for 3 years now. A native of California, Dr. Aiello obtained her PhD in anatomy at the University of London. She spent some 30 years on the faculty of the University College London, 6 as the head of the Department of Anthropology, and 3 as the dean of the graduate school. Leslie is renowned for her breadth of her research and especially for her multidisciplinary approach to research questions. Her research focuses on aspects of brain size scaling, allometric scaling, and the metabolic costs involved in being human. In a word, the energetic costs of being human at different stages in our evolution. Dr. Aiello is widely published with more than 50 articles in prestigious journals such as the Journal of Human Evolution, American Journal of Physical Anthropology, Nature, and Science. Finally, our last speaker this afternoon is Professor Dan Lieberman, who's professor of biological anthropology at Harvard University. He obtained his BA in 1986 from Harvard and a Master of Philosophy from the University of Cambridge a year later. He returned to Harvard where he obtained his PhD in 1993. Thereafter, Dr. Lieberman was an assistant professor of anthropology at Rutgers University from 1995 to 1998, and then an associate professor of anthropology at George Washington University up until 2001, when he once again returned to Harvard to take up a full professorship there. Dan's work is characterized by its diversity of approaches to answering a common question about the apparent uniqueness of the human skeleton. He has undertaken a series of elegant experimental developmental studies on primate craniofacial growth and development, experimental studies on the biomechanics of chewing and locomotion, 2 of the things that seem to set us apart from our closest fossil and living relatives. Dan has already published 1 edited book and over 65 research articles in journals such as Science, Nature, and PNAS. One of these articles, a seminal work on the evolution of human endurance running, will be the focus of his talk this afternoon. Not bad, 7 minutes. Without any further ado, I'd like to ask Professor Blumenschine to come up and to deliver his talk to you on Landscape Perspectives on the Archaeology and Palaeoecology of Early Homo in the Paeleo-Olduvai Basin.
Thanks, Fred. Good afternoon, ladies and gentlemen. Thank you, Fred, for the introduction, and Richard and the rest of the organizing committee, thank you for the invitation to participate. This title was perhaps more appropriate for the workshop, and so I came up with something a little more punchy that might convince you that you shouldn't go to sleep at this point. Carnivory, Stone Tools, and the Origins of Human Distinctiveness, a View from Olduvai Gorge. I believe it was Richard this morning who said that humans are quite peculiar creatures, and odd indeed we are. And paleoanthropology provides a means, and the only direct means, to investigate not only when our distinctive characteristics arose, but under what circumstances they arose. The archaeological record, which begins about 2.5 million years ago, is particularly well suited to this task because compared to the relatively rare hominin body fossils, stone artifacts and butchered animal bones are quite abundant and can be found in a variety of different landscape contexts. And so when we put this archaeological record into a detailed paleoenvironmental or paleo-landscape context, it gives us the opportunity to understand not only the nature of the behaviors that characterized the earliest members of the genus Homo, but also, and more importantly, the circumstances under which these traits were selected for. Now, our distinctive characteristics can be defined by looking at our closest living relatives. Without belaboring this, human locomotion is unique compared to that of other primates. In terms of brain size, this 3 times larger brain in humans than in chimpanzees is something remarkable evolutionary development that needs to be explained. While various primates and other organisms have an impressive tool repertoire, it is only humans who have a dependency on technology for survival. A number of primates consume meat and other products from small animals, but it is only humans who consume animal foods from species the same size as, if not considerably larger than ourselves. And indeed, it is quite odd that a primate, ourselves, is now the alpha predator in most terrestrial and oceanic ecosystems. Of course, we had the discussion of language and communication. We have many common features of communication with our closest relatives, but the language does seem to set us apart. And to pick a more recent development, the fact that we produce our own food and live in stratified societies, not completely unique in the animal kingdom, but unique to primates. And so we can ask the question, when and why did these traits arise? Here's a much more simplified phylogeny than was being depicted this morning. We see that bipedalism and is a very early development, food production a very recent development. I did cheat. I had language up here, but in deference to Professor Tobias, I moved it down a little bit during the lunch break. And then and then we see that there's a suite of traits, the beginnings of the encephalization that characterized the evolution of the Homo lineage, the invention of flake stone tool technology, and this large mammal carnivory, all of which can be viewed as potentially part of a single adaptive complex, as Glenn Isaac and others have emphasized, and which can be associated with the earliest member of the genus Homo. We don't know that for certain. As Richard pointed out, it's possible that there were multiple species, for instance, involved in at least the use of the tools, which is why I often use the term Olduvai hominins when I am discussing this early adaptation around 2 million years ago. Now, perhaps the best place in the world to investigate the behavior and ecological circumstances under which the distinctive behaviors of Homo habilis arose is Olduvai Gorge. This is located in the southeastern part of the Serengeti ecosystem. Olduvai is extremely important, primarily because it has the most complete record in the world of the last 2 million years of geological history, with the oldest deposits dated about 2.03 million years ago, right up to recent times. It was, of course, the work over more than 5 decades of Lewis and Mary Leakey which established Olduvai as one of the premier sites in the world for the investigation of human evolution. We saw this slide already this morning, quite triumphant, having finally discovered a prehistoric hominid at Olduvai in 1959. What Lewis and Mary Leakey did was no less than, for the first time, to document the sequence of change through time over the past 2 million years in the vertebrate faunas in sub-Saharan savanna woodland African systems, to document the nature of technological change during the whole of the Stone Age for human evolution, and of course the discovery of the important fossil hominids, which again for the first time gave us a good idea of the sequence of different hominid forms and demonstrated the coexistence of early Homo with the later Australopithecines. The early part of the record at Olduvai from bed 1 and lower bed 2 is perhaps the richest part of the record. The stone technology here is the Olduvai industry, and the Olduvai, of course, named after Olduvai and the work that Mary Leakey did in defining this type series for the earliest stone tool industry. The functional capabilities of this industry are several. First of all, and perhaps most importantly, the invention of a slicing technology which gave hominins access to foods from larger animals, particularly flesh. The possession of chopping-like tools with jagged edges that can be used to chop, and also the use of unmodified stones for pounding purposes, which gave hominid access to a variety of resources, including the fat-rich marrow from the limb bones of relatively large animals. Associated with these Olduvai stone tools at a number of sites at Olduvai, as represented by those excavated by Mary Leakey, are fragmentary animal bones that are thought to represent food refuse of hominins. And as well, it was Mary's excavations first at FLK Zinjanthropus and later at several other sites in bed 1 and lower bed 2 of Olduvai, which for the first time allowed us to see the spatial arrangement of stone artifacts and associated fragmentary animal bones, and to begin considering the social context of Homo habilis activities. The importance of carnivory to Homo habilis is extremely well established. For example, here is the proximal ulna of a wildebeest-sized animal that has clear stone tool cut marks on it, which is indicative of defleshing and in some cases of disarticulating bones. We also see, as first recognized by us in 1989, the presence of percussion pitting on a substantial portion of the bones from some of these sites, which are, again, incontrovertible evidence that hominins were taking advantage of the calorically dense resources that can be found within bones of relatively large animals. Now, traditionally, the man-the-hunter paradigm during the 1960s and 1970s was the dominant explanation for how hominids acquired these animal foods and how the selective pressures associated with with hunting would have given advantage to individuals with capabilities toward tool use, manual dexterity, etc. One can, however, and I was one of the people to begin to question this man-the-hunter paradigm and to wonder if instead hominids may have been acquiring these foods from large animals through scavenging of various types. As well, we can see, in addition to cup marks on bones, we also see tooth marks on bones made by large carnivores. This is direct evidence of some sort of competition between hominins and large carnivores. In other words, direct evidence bearing on the nature of the Olduvai hominin encroachment on the larger carnivore guild. And so this is when our rise toward now being alpha predators in most ecosystems starts, and I think is a good way to define a grade of hominin adaptation, as Bernard Wood was talking about this morning. The question becomes, were hominids directly competing with these carnivores through confrontational scavenging, or was the competition more indirect, with hominids passively scavenging abandoned carnivore kills? But we do know they were competing. The stone flakes put them in a position to compete with the flesh specialist carnivores and the larger pounding stones with the bone cracking carnivores. One can, as I have done, look at modern scavenging opportunities in the Serengeti National Park. Wonderful way to spend a year. And try to understand the ecology of scavenging. Basically, how to obtain a scavengeable meal of fresh animal foods without becoming a meal for a carnivore yourself. And there are a number of opportunities, but these have different advantages and disadvantages associated with them. In open habitats, for example, cheetah kills or kills made by hyenas, competition for these carcasses with carnivores is high, as is the risk of becoming prey in scavenging the carcasses. A very high-yielding opportunity is drowning deaths, yet such drownings are relatively rare in modern systems. And on the other hand, in woodland settings near rivers and lakes, one gets lower competition for carcasses with the availability right at hand of an arboreal refuge, something we have good evidence that Homo habilis was capable of taking advantage of. And so tree-stored leopard kills, as well as abandoned lion kills and kills possibly abandoned by saber-toothed cats, would have provided regular, predictable, and fairly high-yielding scavenging opportunities. We can test this scavenging, passive scavenging idea by looking at the proportion of bones that are tooth-marked by carnivores in different, when we vary the order of hominid and carnivore access to carcasses. And so when there's no hominid access, when, for instance, hyenas break up long bones by themselves, over 80% of the resulting fragments come to bear recognizable carnivore tooth marks. Conversely, when hominids first remove flesh, break bones for the marrow, discard this material, hyenas will come and selectively scavenge remains such that the overall frequency of tooth mark bones is much lower, around 15 or 12%. This hominid followed by carnivore model is a stand-in for expectations of what we should find if hominins were hunting or scavenging in a confrontational manner. We can alter the sequence, as my student Marie Salvaggio did last millennium, and look at what happens when carcasses are defleshed by felids like lions or leopards, followed by hominid breakage of the bones and removal of flesh scraps. This is modeling passive scavenging from felicles, and we see distinctive frequencies of tooth marking much higher than the hunting model of about 54%. And finally, we can look at, for example, the bones from FLK Zinjanthropus, which excavated by Mary Leakey, and we see that the frequency of tooth mark bones here is most similar to and indistinguishable from the mean for the passive scavenging model. Now, a recent paper that came out earlier this year claims that I had confused many tooth marks for what are called biochemical marks inflicted by various microorganisms, such that the actual carnivore tooth marking rate is what you expect more of the hunting model. However, we have looked at this literature now. It's referred to as microbial bioerosion, and you can see that the bioerosion bears no resemblance to carnivore tooth marks. Well, so given the strong possibility that hominids were passively scavenging, we can now try to look at this activity in paleo-landscape context and try to see the ecological context in which scavenging was possible and when it was not. And we can also look at the important matter of how the hominids were able to get the stone tools to the carcasses, for the landscape distributions of these two critical resources is often disjunct. This has been one of the foci of work that I have done along with Fidelis Masao and a large team of scientists since '89 at Olduvai Gorge. Instead of looking at change through time, we are looking at variation in environments and the archaeological record through space in exposures that span an area of approximately 130 square kilometers. There are two target horizons in particular, both of Oldowan age, lowermost bed 2 and middle to upper bed 1. We have put in a large number of trenches over 13 seasons of excavation, yielding a large artifact and bone sample. The distribution of these trenches is ideal because in the prehistoric Olduvai Basin, the center of it was occupied by a lake, the perennial portion of which was about 10 kilometers long, and it expanded to over 3 times the size during wet climate phases over a broad lake margin zone, and then we have deposits also of the alluvial fan leading up to the volcanic highlands. And so the trench sample that we have is from a number of different environmental settings, which allows us to investigate the extent to which variability in the archaeological record co-varies with paleo-landscape setting. We have along the way found some hominids, 12 in total. This is the most important one, which Richard Leakey discussed briefly this morning. We have found tremendous variability, basin-wide variability in the stone artifact and bone assemblages. For instance, here is the weight density of stone artifact assemblages over an east-west distance of 18 kilometers. And the density of artifacts varies over 4 orders of magnitude. Clearly, one hopes that there's a behavioral signal in this variability. Well, one signal we would expect to see if stone tool procurement incurred a significant transport cost to get the stone to where the carcasses and other resources requiring stone tools were found, that one would expect to see a number of changes in the artifact assemblages as distance from one of the major material sources, Nibor Soit, increases. The further away one gets, one would expect lower quantities of the material, lower proportions of the Nibor Soit quartzite relative to artifacts made from other materials, etc. And in fact, we have found that these predicted relationships hold for the sample in the eastern basin, where here we see the weight density of quartzite artifacts decreases significantly with increasing distance from Nibor Soit, reaching near extinction levels some 4 kilometers from the source. As well, the weight proportion of quartzite artifacts shows the predictive negative correlation with distance from Nibor Soit, again, reaching very low percentages beyond 3 kilometers from Nibor Soit. The mean length of quartzite cores, a measure of how extensively the cores were worked to remove flakes, also shows the predicted negative relationship, and within 4 kilometers starts getting down to a size where only bipolar flaking of cores is possible. And so, first of all, the good news is that there is predicted behavioral patterning in the landscape stone artifact assemblages from Olduvai. We haven't wasted our time over 13 years of excavation. As well, stone material transport costs over relatively short distances appear to have constrained hominid land use. However, distance from material source accounts for less than half of the variability in the measures of artifact assemblages that we examine, which therefore suggests that a variety of ecological factors, especially those theoretically related to landscape variations in tree cover abundance and predation risk, had perhaps a greater influence on hominid land use. And here, just speaking simply, one would expect hominid activities to be focused in areas with relatively dense woody vegetation, offering refuge during travel or during other activities, refuge from predation, and conversely, more open areas, exposure to predation risk would be much higher, such that hominid activities should be more limited there, with materials being transported from the open, dangerous locale to the safer places with arboreal refuge. Now, testing these ecological influences, theoretical ecological influences on hominid land use, is complicated by the tremendous environmental variability which characterize even the relatively discrete time intervals that we are sampling. For instance, lake level fluctuations, which seem to be in phase with in a cycle at a lower frequency than Milankovitch cycles. These lake level fluctuations would have inundated broad areas of the lake margin for some period of time, and then during drier periods, the lake margin would be re-exposed. As well, more recently, we have discovered that Mount Olmoti, which was the active volcano during the Oldowan at Olduvai, erupted catastrophically, perhaps as many as 8 times during this time interval, essentially destroying the vegetation in at least the eastern part of the Olduvai Basin and forcing vertebrates, including hominids, to abandon the basin. what would follow then would be a landscape succession. The question is whether the geological record is fine enough for us to detect the nature of this succession, including the nature of hominid recolonization of the central Olduvai Basin. And finally, not only is there environmental variability over time, but also we have discovered a tremendous source of environmental variability over space. When one looks at most lake margin areas in Rift Valley lakes today, they tend to have the very gentle gradient and flat terrain, otherwise flat terrain, and fairly featureless open grasslands. Yet we have discovered that there were a number of faults that were active during this here lowermost bed 2, which would have imparted minor topographic variability to the lake margin zone, and therefore, hypothetically, a rich mosaic of different vegetation types. On the upthrown parts of the faults, we would expect, based on modern analogs that we see in modern protected lake basins today in Tanzania and Kenya, we would expect to see relatively dense woodland compared to the lower lying areas, which would be more open and indeed marshy. And we would expect, therefore, that the hominid activities would be focused in the relatively safe parts of the landscape, and that any activities in the lower lying open parts would be quite brief, with materials being transported to the safe locales. So we are in a position here to try to test the extent to which exposure to predation risk and hypothetically tree cover abundance would have constrained hominid land use. First of all, we see that some mysterious patterning, previously mysterious patterning in the stone artifact assemblages over this some 4 to 5 kilometer stretch seems to be explained in part by the presence of these sedimentary faults, such that in areas on the high parts of the faults, we get greater technological diversity, relatively high proportions of cores and manuports, as well as flakes, while in the lower line, more open and dangerous areas, we see that these tend to be dominated more by the flakes, with the cores and manuports occurring at low levels, as if these, if they were ever introduced to these open areas, were transported back to the safer spots for fuller use there. So to test this predation risk idea, we can use a proxy based on my and now other people's work, which is the percentage of long bone ends to shafts among larger mammals. Basically, when carnivore, especially spotted hyena, densities are quite high, they tend to destroy a large proportion of the long bone ends, leaving mainly long bone shafts. Hyena densities are highest in more open areas of the landscape, that is, those which we have modeled to be more dangerous for hominins. So we can use this fossil index as some measure of the degree of predation risk to which hominids were exposed, and relate it to the weight density of artifacts. And what we see is the theoretically expected positive relationships, not quite significant there, although we've now increased our samples, significant for the inland sites, where the densest assemblages, that is, those presumably where activities were concentrated, occur when predation risk was relatively low. And as well, we see that the functional diversity of the artifact assemblages, indicating that perhaps the range of activities that were conducted, is very low in areas where predation risk is high, and conversely quite high where in relatively safe parts of the landscape. And so we are beginning to see, with a much more refined understanding of the spatial and temporal variability in environments, as well as our models that predict the nature of hominid activities in different landscape settings, we are beginning to see that this broad-scale landscape archaeological record is informative about some of the circumstances under which some of these novel human traits which appeared in early genus Homo arose. So we can conclude that stone tools allow hominids to encroach on large carnivore niche space. Nothing terribly new here, just said in a slightly different way. Hominid activities, however, remain constrained by predation risk. And so, although hominids had now entered the carnivore guild, their activities were still very much constrained by these very large, dangerous predators. As such, confrontational scavenging does not seem to have been a realistic mode of carcass acquisition for hominids of this time, so that scavenging from freshly abandoned felid kills it can explain what we know about the animal bones associated with the Olduvai stone tools. And finally, the costs of stone transport and the metabolically expensive brain, to anticipate part of what Leslie Aiello will be talking about today, may have been offset by calorically rich animal foods. And it's particularly the youngsters here with marrow, fat-rich marrow, who have the most expensive brains, that this scavengeable food may have been most important to them as weaning foods. Thank you very much for your attention.